Okay, welcome everybody. It is 5:30, so I figure we'll get started. I want to thank you for coming to Moss either earlier today or if this is your first time. Welcome to the second half of the Misericordia Ocean Sustainability Symposium. Welcome to those of you who are streaming in online, as well as those who are present today right on campus. So before we start the speakers, I want to say a few words. I wanted to thank all the students who are involved in Moss today. There were 6 classes who were involved and who showcased their research and work that they did earlier this all throughout the semester. Some students started as early as the beginning of the semester, some about halfway through. So there were lots of hours and hours and hours of work went into what the students have done. And I'm really proud to see the work that the students put on. There was a lot of effort made. And it's very exciting. This is the first, Ocean Sustainability Symposium that we've had. And I'm really excited to talk about that. One of the reasons is I didn't introduce myself. Most of you know who I am, but if you don't, I am Dr. Linda Auker. I am the Program Director of Environmental Studies and Assistant Professor of Biology. And our Environmental Studies program is in its 2nd year. So we're relatively new, but we're off to a great start. We have lots of really enthusiastic students doing good work. And I also wanted to give special thanks to Dr. Cosima Visa, Dr. Patrick Danner, and the Misericordia marketing team, Sam Jarvis, Caitlin Hall especially for their work and getting the word out. Over the last few weeks, we've had a lot of publicity about this, so it's really exciting to see that. So a lot of planning, organizing, getting the word out is very helpful. I also want to give additional thanks to our speaker, who I'll be introducing in a moment, Dana Morse from University of Maine Sea Grant, for joining us as one of our speakers in a discussion about biofouling. So he'll be presenting here in a few moments. And if you missed the earlier half of Moss and you didn't see all the cool things our students did because, we had classes and stuff this morning, or you may be tuning in from elsewhere, I will be posting all of this stuff that has been done on our website. So you'll be able to see a collection of the work the students have done, and it will be also featured in coming weeks on our social media. So stay tuned for seeing all the great things that have been done here. So why an ocean sustainability symposium in Northeastern Pennsylvania, right? A lot of people ask me like, oh, how are you a marine biologist in Northeastern PA? Well, honestly, it's not that hard because we're not terribly far from the ocean. And, when all is said and done, this is where, of course, we are here in Dallas, Pennsylvania. It takes about 2 hours to get there, but it's still takes a lot longer to get to my actual study sites up in Maine and Rhode Island, but it's still a nice drive. But I wanted to give you a few reasons why you should care about this kind of stuff if you didn't already know. Besides the fact that I'm a marine biologist, so of course we're going to have something related to the ocean here. But seriously, we live in the Chesapeake Bay watershed. A lot of the research that I saw today that students have done have focused quite a bit on the Chesapeake Bay watershed. We had a student actually talking about mine drainage into the Susquehanna, which leads into the watershed. We had another student talk about sea bass, the sea bass fishery and regulations in Chesapeake Bay. So this stuff does hit close to home. Chesapeake Bay is also the site of dead zones, much like you see in the Gulf of Mexico, where you have these anoxic locations, very low oxygen, and that's due to eutrophication, which is essentially nutrient pollution. And those different sources, including fertilizers from farmland, industrial pollution, things like that. And they create these big algal blooms, which when those algae die, they decompose, which actually depletes the deeper waters in the estuary of Chesapeake Bay of oxygen. Pollution, of course, is also a concern as the Susquehanna moves through areas that either once were mined, still are mined, go through industrial areas, go through farmland. But, therefore we have an impact on the ocean, but we also benefit directly from the ocean in many different ways, right? The ocean's a major source of food. Even if you don't like seafood, your toothpaste has carrageenan in it, which comes from Irish moss, a type of algae. A lot of ice creams do as well. So a lot of the products you use, whether you're aware of it or not, have materials in them that come directly from the sea, that are sourced from the sea. And of course, we live in northeastern Pennsylvania, which we have a lot of seafood places around here. Cooper's Seafood is a very well-known one if you've ever seen The Office. A lot of people celebrate Christmas and the holidays by buying a lot of seafood. So it's something we do consume quite a bit in this area. Sources of medicine, the ocean is also a major source of medicine, believe it or not. There are a lot of species, like certain sponges, which produce chemicals that can be used as anti-cancer drugs. There are some sponges in the Bahamas. I just told my marine bio class about this recently, so those of them who are here know about this. But Harbor Branch Oceanographic Institute in Florida has studied several species of sponges that live on coral reefs that produce chemicals that have anti-cancer capabilities, specifically anti-pancreatic cancer, right? So, obviously that's something we all should be interested in. And in oxygen, most of the oxygens produced through photosynthesis doesn't come from trees. It comes from the phytoplankton in the ocean. So the ocean's health is critical for our own survival, right? We also benefit from the ocean's role as a carbon dioxide sink. A lot of the carbon dioxide in the atmosphere dissolved in the ocean. Now, of course, that comes with its own problems. When you have excess carbon dioxide in the atmosphere, it actually reduces pH levels, increasing acidity in the ocean. And that's something else some of my students talked about today in Moss. And of course, we're thinking about recreation. We vacation at the beach, near or far, whether you go down the shore, right, to Atlantic City or Delaware or Ocean City, Maryland, or you go up to New England, or you go over to Hawaii, you're enjoying the ocean, right, and all the things that it has to offer. So there's many, many reasons why, even if we're in landlocked northeastern PA, we should really pay attention to ocean sustainability. So I want to turn it over now to Dana Morse, who is a senior extension program manager and aquaculture lead at Maine Sea Grant. He's based at the Darling Marine Center in the University of Maine. And he's going to talk about some of the research that he and I and many others have been involved in over the past few years, thinking about one of the major resources in the ocean is aquaculture, right? And one of the things that is commonly aquaculture, that came out weird, but one of the things that's commonly farmed in New England are scallops. So he's going to talk about scallops. He's going to talk about something called biofouling. And then I'm going to come back and talk a little bit about some of the tools that we can use to monitor for biofouling and communicate biofouling data. All right, so Dana, take it away. Hopefully you can share your screen.
Hi, thank you, Linda. It's great to be here. And whoops, I'm immediately interrupted by my dog. He was trying to get in the room. I do have a question. I can only see myself and a screen. Should I be seeing the audience as well?
No, you should not be.
Okay, well then I am going to share my screen here. Thanks again for having me. I've enjoyed working with Linda so far and looking forward to more. As Linda said, I work for the Sea Grant program. I also work for the University of Maine Cooperative Extension program, and I'm a quote unquote extension agent, which means I'm kind of a middle person between industry, science, academia, regulatory, and lots of goods and services. And for those of you who might be wondering where you're headed in your work career, I'm one of those people who can say without any trace of sarcasm, I love my job. It was one of the things that I've been most lucky at. So it is possible to be in the marine world and find a job that you truly love, and I've been at it since 1998. So if you're wondering, well, take hope. There's good stuff out there. So the stuff that I've prepared here this evening, and I'll try to go, I don't know, 20 minutes, 25 minutes, so that we can have a little bit of time for discussion, is all about biofouling. Here we have a couple of photographs of biofouling, and if you let me, I will talk about scallops all evening long. But I'm gonna hold back. But both of these examples are scallops. On the left-hand side, this is taken up in Nova Scotia. It's a series of containers called pearl nets, and they all have scallops inside. But what you see on the outside is a complete covering of things called sea squirts. And then on the right-hand side, you can see the shells of some scallops that we've grown at the Darling Marine Center. And they're covered mostly with macroalgae, but you can see lots of other things in there. And biofouling, in case you're not familiar with it, is the stuff that grows on everything that you leave in the ocean. And if you leave a piece of wood or a piece of metal or a piece of fiberglass or an oyster or a mussel or a scallop, something in the ocean is going to want to settle on it and grow. And this turns out to be important for the aquaculture industry. Backing up quickly, and briefly here, the Sea Grant program, in case you're not familiar with it, is a national program. Every state that has a shoreline, including the Great Lakes, has a Sea Grant program. We fund research, we do education, we do outreach and extension and communications. And these are my colleagues up and down the coast. We're based out of the University of Maine at Orono. I live in the mid-coast, but I've got colleagues who live 4 hours from one another. And so we all kind of travel the coast as part of our work. And so here we are. I know I can't see you, so I won't ask the question. But if we were live, I'd say, well, where the heck are we now? And what you're looking at, of course, is the Earth. This is the Pacific Ocean. And what you're looking at the sort of the 1 o'clock position there is New Zealand. And so this goes to Linda's point about, well, the ocean is kind of important. Every second breath you take is coming from oxygen from phytoplankton. It's a source of major climate influence, and obviously it's a major source of food. I just love this. I was playing around with Google Earth one day and spun the Earth, and of course, nearly the entire view is water. So that gives you a pretty stark view of what we live with here in planet Earth. It should have been called Planet Ocean, really. And the next thing that I'll say is that this talk is pretty well timed, because this document, which was produced by UNESCO, just came out last week. And I offer it, including the URL, and I'd be happy to send it to you as well. I offer it for two reasons. One, because it's relevant and it's recent, and two, because this goes to the point of biofouling being really important in all kinds of marine industries. And so we're not talking about just some mussels on your oyster cage, even though that's the context that we're using today. This is something, for example, that the Department of Defense spends millions or billions of dollars on, because obviously boats accrue biofouling. And so understanding biofouling and the mechanisms and the organisms is really important. For us as producers, and I do participate as part of the industry. I'm a leaseholder in the state of Maine. My farm produces oysters. So we all have to deal as farmers with biofouling of different types. And these are the reasons why we care and why we should care. So first of all, I'll touch on the money aspect of it, because every aquaculture operation, not every, but most of them are businesses. And even though people get into the business because they want to be their own boss, they want to work outside, at the end of the day, to be able to keep doing that into the future, you have to make a dollar. And so aquaculture businesses are farming businesses. And as you may already know, labor is a huge expense in the farming industry. And that's no different in aquaculture. And so when you have to deal with biofouling, that equals labor and that equals money. And so you can track that right to the bottom line and the profitability of your business. So money's important. Environment is important. I came across this almost ridiculously laughable photograph in this UNESCO report. And there are lots of issues related to environment with respect to biofouling, most of all, or perhaps principally, invasive and non-native species, much of which comes through shipping. Not all of it. Aquaculture has certainly been guilty and implicated in the transfer of non-natives, but the shipping industry is definitely a principal driver of the transfer of species. And I love this photograph because it's both. Here we have a ship that is going probably a fair distance from one place to another, and the thing that it's carrying is an enormous aquaculture pen. So I kind of got a kick out of that particular photo. And so here's me on my farm, all bundled up. This is probably like last November or so. And the last bit, and not the least important either, is ourselves. If you've ever worked on a farm for any length of time, you will know that it is tough on your skeletal system, on your muscular system, sometimes on your circulation, all kinds of things. And so if you want to be in the aquaculture business for a long time, you have to think about taking care of your body. You have to think about ergonomics. And even if you're not practicing a yoga pose there on your dirty, muddy boat, when you have to spend time dealing with biofouling and spending a lot of time holding a pressure washer or flipping a cage, that's going to take a toll, especially as you get older and especially later in the season. So biofouling is important from a lot of perspectives. I wanted to switch direction a little bit and just give you a little bit of context. The aquaculture industry in the state of Maine is about $90 to $110 million farm gate value. So that's right off the farm. Most of that is Atlantic salmon farming, but our principal other species, eastern oysters, blue mussels, and seaweed, are definitely getting up there. I list out sea scallops that are about $200,000 a year. Hard clams and soft shell clams are less than that. I don't have a good graph for seaweed, but it's virtually asymptotic. The rise of seaweed since, say, 2015 or so has been quite rapid, and it projects to continue to be rapid. I put the chart in here to show the context of the aquaculture industry versus our other major marine commercial industry, which is the lobster industry, which in 2021 landed upwards of $700–800 million worth. That's a lot of money for 4,000 or 5,000 lobstermen and women in the state. So aquaculture is a minor part of the overall pie here, but the danger to a pie chart like this is that Maine is over dependent on lobster as a resource. And with climate change, there are a lot of people who are very nervous about this, as well as legal challenges with the right whale issue. We don't have to get into that, but I'm happy to if you'd like, and wind development. So the lobster industry is a huge driver of economics in Maine, but it's also uncertain. And so aquaculture is one of those things that seems to be filling a need. There is no shortage of things that act as biofoulers in Maine and elsewhere. And again, of course, I have to show you a couple of sea scallop photos because I work with scallops and I'm completely enamored of them. The two photos show damage by a thing called the boring orange sponge, or the orange boring sponge. And I don't know if it's boring, but it does bore. This is Cleona. And you can see through the light there that the boring sponge can drill completely through a shell. and it's kind of pretty, but perforating a shell is hazardous to the shellfish because it has to spend energy, and it makes it much more susceptible to predators. If a crab were to come up and squeeze this shell, it would just crumble. And so I've given you a list here. There are a variety of genera in here, and they all come with their own life histories, their own challenges, and their own methods by which farmers try to deal with them. I wanted to show you a few photographs just to kind of give you a sense of the major species that we're growing and some of the typical culture techniques. This is kind of a typical oyster farm shot where you've got these plastic mesh bags that are held afloat on the surface by floats on the side of the bag, and those are the white styrofoam billets there that you see. And the oysters are in the bag, so they're nice. They're in nice warm water up at the surface. But of course, on the underside of the bag is where the biofouling starts to accumulate. And so every week or two, the farmer will come along and flip each individual bag so that the stuff that is now in the water becomes exposed to air. It eventually dies off and kind of dries out and disintegrates. And in the next photos, you'll see this quote unquote air drying approach replicated, because really you don't have to spend money on a power washer, for example. You do have to spend some labor to flip the thing, but at least Mother Nature then does the job. And these units are sort of the next step up in scale from that. These are called oyster grow units or oyster ranches. And in the right photograph, you see the wire mesh cage, and those have the oyster bags in them. They are flipped up for a day or two so that they can dry out and the biofouling will die off. Most of the time, these oyster cages are as positioned as they are in the top left, where the floats are holding the cages at the surface of the water. And it's nice and warm up there. There's plenty of feed. The farmers can get to them for harvesting and that kind of stuff. So here's a system that took the air drying approach, and they made a whole system out of it. And globally, it has become a very popular system. This, I'm going to show a little video. This is a system from New Zealand called the Flip Farm system. And I offer this partly because it's a novel take on air drying, but also because it illustrates that aquaculture is a global industry, and this is technology from across the planet. And so what you see are these lines of cages. The ones that we're looking at now are air drying. But you might imagine that once they're air dried, I'm going to go back and start it a little bit here. The ones on the left of the screen are ones where the oysters are in the water. And there's a nice little doodad that you attach to the side of your boat, and you just zip up and down these lines. And a single person in a boat can flip thousands and thousands of these cages a day. So it's highly efficient. So this is relatively new technology in Maine. It's been here about 3 years or so. Continuing the air drying approach, this is also technology or a system from elsewhere, really kind of pioneered in Australia and in Alaska. This usually happens at the lower intertidal area that in Maine, we have about anywhere from a 9 to a 25-foot tide. And so at low tide, when these bags get exposed, that means they kind of dry out. And what's more, sometimes these bags can have floats on them so that at high tide, the bags will kind of flip up, and at low tide, they'll flip down. So that keeps things shaken up, and it helps to keep things clean. So there are some of the kind of, I think they're kind of ingenious ways that farmers use to combat biofouling in oysters. I'll switch gears for a minute to cover mussels. And as you may have seen, if you put a line in the water, if you put a rope at the surface of the water anywhere in Maine long enough, you're going to get a clump of mussels out of it. And so what mussel farmers do is that they have special lines that have pegs in them, these yellow pegs, and they'll bundle this line up at the surface, and they'll hang it out there for 3, 4, 5 months. And then when they pull it back, you see all those little black dots on there. Those are seed mussels. And so collecting mussels from the wild is very standard. the next thing that happens, and I'll hope this works, is that they essentially make a sausage out of it. And what you're looking at there is a hopper by the guy in the tank top there, and the hopper has the seed mussels. There's a rope that goes through that machine, and then there's a cotton mesh, the stuff that's spinning around in a circle. And you can see this long sausage of mussel seed, then gets walked out and it gets hung onto a raft. And this is a pretty labor-intensive process. And the mussels themselves will start to grow, and they'll either grow through the mesh, or really the cotton mesh on the outside simply disintegrates. And so what you're left with is a rope that has a bunch of mussels clumped onto it. And they're usually hung from a rope, from a raft like this. And those ropes might be 50 feet long. And it's not so much the mussels themselves that get fouled up, but if you look at the raft, you can see some green netting that's wrapped around the raft, and that netting will go down 50 or 60 feet, and that's because eider ducks will eat mussels. An eider duck weighs maybe 5 to 7 pounds. It can eat half its weight in mussels a day, and if you have a flock of 5 that come in one day, you'll have 20 the next day, and you'll have 100 the next day, and you'll have 500 the next day. And there are more than one stories of 40,000 pounds of mussels being eaten from a raft in a week. So farmers use these predator deterrent nets to keep the ducks from swimming in and eating all the mussels, but the nets get dirty. And they get really heavy, and it's probably 30% of operating costs that mussel farmers have to deal with to keep this stuff clean. Here are some photographs from Prince Edward Island. They use a little bit different system, but you can see in the right, you get a whole bunch of these sea squirts or tunicates on there that can smother mussels. And if it doesn't outright kill them, it can certainly slow their growth and turn everything very heavy. And in the bottom, you see some starfish, and starfish can kind of be considered biofouling. And if you let starfish on there long enough, they're going to eat all your mussels too. So you want to have a method to get the biofouling off. And the Canadians have gone to great lengths to control this. So what you see here is a big engine on the right-hand side with a great big pump. And on the left-hand side, that metal thing opens and closes. It's like a walk-in closet. And they will lift up their lines. They'll put their line into that closet, and then they'll blast it with water. Just a huge expense in equipment and in labor. And as you can imagine, if you have to do this with thousands of dropper lines, it's going to affect your bottom line. And I don't want to leave seaweed out here. Here's a line of sugar kelp on the left and dulse on the right. The seaweed industry does have to deal with biofouling as well. And here is one case. These are the snails called the kelp snail, Lacuna vincta. And in the middle of the kelp growing season, so around March, April, and maybe into May, the lacuna settles onto the kelp, and they start laying their eggs. And they're these little sort of donut-shaped egg masses. They're maybe 1/4 of an inch in diameter, and they kind of spot the kelp. And since most of this kelp is going to food, kelp farmers don't want their product covered with lots of little snail egg cases. So they have to deal with it as well. I've got one other shot to follow this. They can wash it off, but this one here is membranipara, and you can't really wash it off. It's a bryozoan, and once it attaches and starts to grow, you really have to cut it off. So now seaweed farmers who might be dealing with 50,000 pounds of product, they have to know something about timing. They have to know something about depth. They have to know something about the biofouling organism so that they can get, they can get their product to market in good shape. And here's a shot of someone washing kelp just for that kind of reason, to knock off egg cases, to knock off any kind of like sediment that might be on there, anything that else might be considered biofouling. So there's extra labor equals money. There are other organisms that affect aquaculture crops in different ways. The black splotches inside the shells of these oysters are caused by something called Polydora webstri, the mud blister worm. And it can kind of affect the health of the oyster, but really what it does is it affects the marketability. Because if you have a lot of these and you go to shuck this oyster, sometimes the shucking knife can pierce that mud blister, and essentially mud and worm poop then leaks out over your beautiful half-shell oyster. No one wants to eat that. So here's the thing that's not really growing on the shell, it's growing in the shell, and yet it kind of qualifies as biofouling. And then I think this is the last bit in the list of why biofouling is important, is boats and other equipment. And people pay a lot of money to keep their boats defouled, either scraping them using copper-based anti-fouling paints, which isn't that great for the marine environment. This is a novel approach called Finsolate, which was modeled after sea urchin spines, believe it or not. And it's been tried a few places, and including in Maine. There was a trial of this last year. So you can imagine the recreational boating industry, the commercial boating industry, that's billions and billions of dollars who pay a lot of attention to biofouling. I'm going to close up here by saying that in this diagram, which came from that UNESCO biofouling document, hopefully you can see it, but there is a whole slew of ways in which people try to keep fouling from happening in the first place or deal with it once it happens. And it is a significant source of, or a significant effort for research and development. It is a significant source of expense. It's a kind of a significant source of business opportunity as well. If you have a new gadget or a widget or an approach to reduce the effects of biofouling, well, then there are business opportunities there. And I'll close by trying to draw this all together and maybe pave the way a little bit for Linda's talk, which is that there's some really cool stuff that kind of feels like a recipe to me. So for example, the hobo pendant that you see there is a $54 temperature logger, which you can put out on your farm. It will log temperature for years. It will Bluetooth to your phone. So it's a really affordable way for Joe or Sally farmer anywhere to get important temperature data. You can combine that with things like the thing on the top right, which is a fouling plate, so that you can get to understand the patterns of biofouling on your farm. And you can access scientific information like circulation patterns or buoy systems, which are producing very fine-grained data about things like chlorophyll, turbidity, wave height, all those kinds of things. And then when you put this all together, here's the recipe part, you can really get some insightful observations and guesses as to what's going on. And it's especially cool in my idea when, or in my mind, when you get growers who might be in a given embayment or an estuary, and they've all got some of these data to share. Then you start to develop very detailed pictures of what might be happening within a river system or within an embayment. And then you can understand things like biofouling or harmful algal blooms or transmission of diseases and invasive species and that kind of stuff. And so all of these things work synergistically, and it's a pretty exciting area. I think I'll stop there. You've seen some of the bad guys and some of the major species in the Maine aquaculture industry. And hopefully we have a couple of minutes in case there's some discussion or some question. Thanks.
Thank you, Dana. You really kind of opened up the floodgates for me to talk here in a few moments. But before we do that, are there any questions from the audience for Dana about what he just told you? Question at all? Yeah.
And we take our boat from obviously to Raystown Lake to Chesapeake Bay, and I know you got to be careful about cleaning off the boat, but we never leave our boat in the water. So I'm just curious, how long could these critters live on your boat and then you take it to another water source?
So the question is, how long do these critters, did you hear the question?
Only sort of. I need to repeat,
Okay.
please.
So how long do the biofouling species tend to live on, say, boats, for example? So if you were to move them from one water body to another, how easily would they be spread?
Yeah, the answer is very easily. If you have a part of your boat that, or other structure, like a raft or a cage or something like that, that starts to gather biofouling, those animals can be on there for a long time. Sometimes some of them can live out of the water for a long time, but certainly if they're immersed in water, you could go from one end of the coast or one end of the coast of Maine to the other, and they'll be right there with you. And so what's happened in Maine, particularly in the salmon industry, but also in the shellfish industry, is a series of protocols to try to limit this. So in the salmon industry, they have a thing called a bay management plan that sets out very definite processes for the movement of gear, for the movement of animals, in this case fish, and for the movement of people. So there are biosecurity measures in place to keep things from hitchhiking on gear. There are biosecurity measures if you're going from one facility or one boat to another to make sure that you don't have hitchhikers on you. And in my case, I send out notices to growers in the state about used gear pretty frequently. And so one of the things that we did was the Department of Marine Resources, the Maine Aquaculture Association, and Maine Sea Grant, we put together some advice, some pretty detailed advice for growers. If you're buying a piece of equipment from a person, here's what you want to do to limit your exposure for a biosecurity problem, transfer of diseases or something like that. That's a little bit of a long-winded answer, but it's something that we definitely pay attention to.
And I know in Pennsylvania, I'm not sure if they have a lot of like boat washing stations if we're thinking about freshwater. I used to live in New York before this, so there were places you could go, but voluntarily to wash your boat. So I'm not sure how well it's regulated here for that.
Thank you.
Yeah, sure. Any other questions for Dana? I have one about the oyster, the little flip-flop thing they did in New Zealand. How new is that?
Let's see. In 2019, I saw it for the first time out on the West Coast. In
Okay.
2020, so I kind of sent this new technology around to growers. Hey guys, you might want to check this out. I think it was 2020, one of the producers got really enamored of it, took it upon himself to go to New Zealand and essentially get his hands and eyes on it. And I think he is at least a regional, if not the United States distributor for this type of gear. I think the first deployment was in 2021, and you're already seeing, or at least I'm already seeing, farms begin to deploy this equipment as a new thing to try. Growers are always looking for the better mousetrap, just like farmers of any type. And so this technology is not 100% bug-free for Maine because we get ice, we get sea ice up here. But there's a lot that is attractive about it. And so people are trying it out.
Okay, thank you. Yeah, the last time I was in New Zealand, which was 2007, I didn't see anything like that. So it was just like, this must be really super new, but that's really cool. Yeah, because I mean, we did a lot of our work was on the mussel farms, and they have like the rafts there. So that's really neat. There's a question in the chat. Can you speak further about what they were putting on the boat in your one picture? You mentioned it being based on urchin spines, or
Yes.
provide a name to Google.
Yes, I can. Thanks for the question. The product is called Finsulate, Finsulate, Finsulate.
Okay.
Yeah.
Finsulate, that's cool.
Yeah, and it had a limited trial. And not to be a complete wet blanket about it, but my understanding was that the trial was not wholly successful. What it does is you have to clean the boat hull, kind of prepare it, and then it's almost like a fabric. And the length of the nap on the little spines on the fabric vary. The trial in Maine was with the short spined one. They may have had a different result with the long spined one. And the theory is that biofouling organisms have a difficult time attaching to those little hairs, those little spines on the fabric. And that even if it does foul up a little bit, it's a lot easier to wash off than if you have to scrape the whole hull of your boat, for example. So I'm no expert. That's the limited experience we've had here in Maine that I can tell you about. But the thing that I love about it was that it showed really creative thinking, and just goes to show that if people got an idea, there's room for creativity here.
Absolutely. And it's always fascinating when they base it off of stuff in nature.
Definitely.
Very cool. Any other questions? Well, Dana, I don't see any other questions in the room, but maybe when I start talking about some of the stuff we've done, that probably might generate some more. So sit tight.
All right, I'll stay by.
All right, let's see if I can share my screen here. There we go. Can you see that? Dana, can you see my screen?
Yes, I can.
Okay, thank you. All right, so I just want to thank Dana again, because he really kind of opened the can, not the can of worms, it's not really what I wanted to say there, but he really kind of like led perfectly into what I'm going to be talking about. So just a little background, Dana and I started working together, I think, I don't know, 2018, 2019, something like that. It was before I came to Misericordia. And because he was looking for somebody who knew a little bit about biofouling and how to measure biofouling. And so I was asked to be a consultant on a grant that he was working on with Dr. Damian Brady and a couple grad students, Sherwin Coleman at the University of Maine. And yeah, we agreed to work together, and that's kind of where a lot of this project came from, and that I'll be talking about today. And it's kind of opened a lot of doors for what we can do with biofouling information. So one of the things that I'm going to talk about, or the thing I'm going to talk about, is using a tool called GIS to communicate biofouling in aquaculture. So in order to do that, right, I'm going to talk a little bit about what GIS is. For those who don't know, some of you do know because you're in my class, or my class that just ended. Then we're going to talk about what is biofouling. And since Dana kind of went into that in a good amount, I'm not going to spend a ton of time on it, but I'm going to, pick out a few things from the literature that might be of interest. And then I'm really going to dig into some of the workflows. The first one is, what are these panels that Dana showed at the end of his talk? How do you deploy them? What do you do with them when you're done deploying them? How to analyze them, and how to use GIS to display the data. So I'm going to show you some of the dashboards I've been working on. If somebody is interested in kind of contributing to this data set or learning how to analyze the panels, there's always that opportunity here. So let's talk a little bit about what GIS is. So GIS stands for Geographic Information Systems. It basically data, which is data that includes latitude, longitude, right? Location. You use that to inform decisions, detect patterns, and make predictions, right? So if you're just looking at a map with a bunch of points on it, right, that is showing you a pattern, that's showing you where things are located. But you can also use that information and tools found in GIS software to make decisions. Like, for example, if you want to build a new business, like, where's the best place to build my business? Well, you might look at the demographics of that area. You might look at who's interested in, purchasing your items that you have for sale. sale, you know, in order to figure out where to build or base your business. And also to make predictions. GIS is commonly used in ecology to figure out, you know, what will happen if, you know, the temperature of the ocean increased by so much, you know, that's projected by, let's say, 2100. Well, how will that impact, say, coral reefs expansion or decline? So you can use GIS tools to look at spatial spread over time. It's a really very powerful tool, and you know, it's got a little bit of a learning curve, but once you learn it, you can do a lot with it. So all of us here use GIS in some way or another. It touches our lives in many ways. You know, if you use your phone to get from point A to point B, those principles of GIS of finding the fastest route from point A to point B, right? UPS and Amazon and all these other companies that deliver all your holiday presents are using it to figure out the fastest, best route to quickly make their deliveries, right? All those COVID-19 dashboards we use to figure out where all the cases are high and low and declining and increasing back in 2020 were built using GIS technology. And then, of course, you know, even if you want to look at where your property line boundary is, Luzerne County, right, where we are here in Dallas, has a GIS portal. You can look at your own property boundaries, see where your property ends, right? And again, that's using GIS technology. So how on earth does this relate to what Dana just talked about? Well, what GIS can be used for very effectively is to take data, put it into some, if you've heard of ArcGIS Pro or QGIS, that's some of the software you can use, and build applications that communicate information, right? And that information can be communicated to the general public, like, again, those COVID-19 dashboards might do. So we can communicate using spatial data the time and location of where this biofouling occurs, right? Which, as you heard Dana say, would be really valuable if you were in a certain embayment, as he said, and you want to, you know, you want to see what is growing in that location, and, you know, you have some maybe environmental data. You can take, you know, the biofouling that's going on, the environmental data, and you can put it into GIS and see maybe how that's biofouling is spreading out from where you are. You can do some really sophisticated predictive analyses and build models using GIS. So I'm going to switch gears to biofouling, and then we're going to put it all together. So biofouling, as you know, a lot of this has already been said here, but if we're looking at this from kind of a biological perspective, we're taking a look at organisms that recruit or go from a larval stage to an adult stage and settle on a surface of a substrate and metamorphose into adults. And these can be species that grow on other species. We call those epibionts, or species that actually live inside or bore inside other species, endobionts. That boring sponge is a good example of an endobiont. Now, how much does biofouling cost a grower? It depends on a lot of things. It depends on how you are growing your species. Now, as you saw, Dana showed a lot of different methods for growing oysters and mussels, for example, and scallops. So how you grow these different species is going to affect how much biofouling you have. It's also going to be region dependent. Where you live, right, is going to determine what species arrive there. But according to some of these sources that I have listed here, costs associated with biofouling can range anywhere from 5 to 30% of market costs. So it can be, you know, can really bite into your profits quite a bit. So fouling organisms tend to be sessile species. So these are species that stay in one place once they settle, right? They're not going anywhere. So think about mussels, scallops, algae, right? Seaweed, those sea squirts we're looking at, sponges. These are all sessile species. but they also have a free swimming larval stage, many of them, which means that's the part of their life cycle where they actually get to move around. And that's the part of their life cycle that's actually seeking a good place to settle, right? So what you can do with this information is you can put out these panels, and I'll show you a picture similar to the one that Dana showed. But these panels, right, give the larvae a place to settle. Now we know that the ocean, as Dana mentioned, if you put a line in the water, a bunch of stuff is going to end up on that line. It's going to grow on that line because space in the ocean is really limited, right? There's not a lot of places to grow or go, right? So these panels kind of provide space for species to settle. So if you put a panel in the water, you're going to get stuff growing on it. So usually what happens is the larvae will settle in the panel and go through metamorphosis and become an adult. So these are some of the species. I mean, Dana gave a much more exhaustive list than I have here. I'm just showing you some of the more common ones. So most of these I've worked with in some way or another. This one's called Didymnum vexillum. That's a scientific name. We have a lot of names for it. Commonly, the carpet tunicate, pancake batter, or even a tunicate from hell, because when it grows, it's an invasive species and it forms these long tendrils. You can see here, it's a colonial species. These tendrils can break off and be swept with the current elsewhere, settle, and form a whole new colony. And this stuff spreads like crazy. In fact, here it is. This is overgrowing mussels in New Zealand. These are overgrowing mussels here in New Hampshire in the bottom. At the top, these are overgrowing the pilings in Rhode Island, Narragansett Bay, Rhode Island, 2004. So here we have even more sea squirts. These are solitary species, where Didymnum was colonial. These are solitary. It's called the vase tunicate. Also grows very abundantly. This is at the bottom of I believe it's the bottom of a boat, where this stuff, it's been a boat sitting in the ocean for a while, and this stuff will just grow and grow and grow and grow. And it's a real problem when it's growing on your mussels, for example, because then you have to pull these really heavy mussel lines out of the water, which are super heavy, and cause some of your mussels to fall off the line. This is one of the concerns of mussel farmers who use lines to grow their mussels, because these things carry a lot of water in them. Seawater can be really heavy in large amounts, right? Here's another colonial sea squirt called Botuloides violaceus, also an invasive species. And this one looks a lot like Didymnum, but it's bright orange. So it's really stands out from some of the other species. And this one really surprised me. It wasn't until I started working with Dana and the team up in the university that mussels were kind of a pest, because since I've been studying them, I always study them as, oh no, they're being overgrown by invaders. Oh my gosh, we need to save the mussel. And then I find out they're a pest on scallops, which really shocked me. So yeah, they're a biofouling species too. And they grow, as we saw in Dana's talk, they grow in large, high abundance when you put lines in the water, right? And so they actually are pests on scallop farms. That's a new thing I learned recently, which is really wild. So as you can see, there's value to monitoring for these biofouling species because they have such a high cost for mussel farmers, right? There have been studies showing that monitoring for biofouling is kind of, early detection is the best way to prevent major problems with these species. So monitoring for biofouling in Australia has allowed farmers, restock after peak settlement, to time when they, take out their cages or replace their cages so that they're either, so they kind of work around the biofouling season. So what we get from this is, as Dana mentioned, you have all these ingredients, right? You have all these data that you can collect. The presence of biofouling organisms, the environmental data, and you can bring it all together and create an informational tool that you could share with others. And that's really what we want to do. This is what I'd like to do. So let me tell you a little bit about how this works in practice. So these are the panels that I was talking about. So these panels are based on panels designed by Dr. Robert Whitlatch, University of Connecticut. I use them for some of my graduate work, shared them with Dana, and he created some. And it's basically a 10 by 10 centimeter PVC panel. And for those of you who are on campus, I have some of these in my lab if you want to see them in person. But they're marked with a grid. 1 centimeter by 1 centimeter squares make up that grid to make panel analysis a little bit easier. And so basically you stick these in the water, you attach them to a line, for example, and you put them in the water and you let them sit for a month, right? And then you take them out. So for the grant I was working with Dana on, they deployed 6 panels, 3 at the surface and 3 at depth, because you might have different species at the surface than at the depth, and that's what we were trying to find out. So when you retrieve the panels, first thing you want to do is you want to bring the panels to the surface. Now, depending on what your setup is, right, you might just take your boat out, pull the panels out, put them right on your boat. Or if you have a scallop raft, like the one shown here, this was on a trip I took in 2019. And Dana and Struan were on that boat with me. And that's one of the scallop farms where they deployed the panel. So you can also bring it up and just set it on the scallop raft. And you take a photo of each plate. So we're interested in how to do that. So then you take your photos, you want really nice photos that are very clear, and then you clean each plate using a knife or pressure washer, scrub brush, whatever you have available. You want to wipe everything off, put it back in the water, and then wait another month to see what ends up on it, right? And then you redeploy the line, put it back in the water, come back in a month, capture it again. So if you do this every month, right, you start to see what species show up at different times of the year, because every species has its own reproductive pattern. So this is what you want to do if you photograph these panels, right? Your photos, when you're taking these, should always be taken at the highest resolution you can. Smartphones are actually getting better and better, as we know. In fact, I have a kind of an old one compared to what a lot of people have these days. As long as you make sure you put it in a waterproof case, you don't drop it overboard, you can use your smartphone. So you want to get the highest resolution you can. So when we zoom in to look at the different species on the panel, we can clearly see what they are. Photos should not have a glare, because if they have a glare, it can make it very difficult to see what's on the panel. It's helpful to number your photos and have a note in the photo, or in this case, we have the person taking the photo has 1 up here. So that's panel number 1. That works as well, right? Another thing you don't want to do is angle the panel away from the camera. You want to get as close as possible, right? So question I ask growers and anybody interested in doing this is, what data do you want from these panels? Do you want to measure just overall biofouling, right? How much biofouling you get on the panel, just to see how that biofouling changes over time. Do you want to look at a specific species? Because as we saw, some species are really easy to just clean off, say the kelp, like Dana showed, you have the snail egg cases, you could just clean off. But if you have the membranipara, which is a really small bryozoan, you can't clean it off the same way. So if you're looking at a specific species that's kind of a pain, maybe that's the only species you care about, right? When you analyze the panels, there are free image analysis programs. I'm going to show you a really quick one that you can use. And that allows you to look at the species abundance over time. Or maybe when you take your panels and you only care about whether or not a species is present or absent, you don't care about how much there is. So you could just say, yeah, there's barnacles here. Yeah, there's mussels here. Yeah, there's sea squirts here. And then that's good too. So it really depends on what kind of information you want to collect. So this is to remind me to show you that software. So the software I'm talking about is called ImageJ. And ImageJ is often used for like, counting cells and whatnot. And you can see that it's not really much to look at. So I'm just going to minimize my windows here. What you can do is you can pull up some panels. So I'm going to grab, these are the panels that were taken in Maine as part of this. Oops, I see my screen sharing is paused. Let's try that again. There we go. So these are panels that Dana and crew took in Maine. So I grabbed a few of these, and I'm going to pick one that I think has, here we go. This is what the panels look like. I believe this one's from Hurricane because those were covered in stuff. But you can see there's a lot of stuff here, right? So, if you want to look at every species on here, you're going to have to identify every species that you see, right? So if you do this kind of work, what you have to do is either you yourself know what species you're looking at, or you have somebody who's really good at identifying species that you can call and say, hey, do this, right? So one of the things that you do, you can analyze this by looking at percent cover. And this program allows you to do that pretty easily. So one of the things that you can do with this, for example, is you can actually set a scale. So I know these panels, I know these panels are 10 by 10. So I can draw a line that's across the panel the best I can. I'm just doing this really quickly here. And you can say, okay, that line is 10 centimeters long, right? And so now the program knows that 1,944 pixels represents 10 centimeters in this photo. So we say, okay. And then what you can do is you can draw, I'm doing this very quickly and very poorly, but you can draw, say, around one of the species. And if you click measure, that gives you the area, right? How many square centimeters that particular species covers. So 6.8% of that panel is covered by Botulotes violaceus, right? So I know the percent cover. So you can go through these and look at the percent cover, do this for multiple, look at it over time and see how the biofouling has increased or decreased with each season. You can also do something really cool with this program, that if I just wanted to say, count how many barnacles are in the picture, there's 1, 2, 3, right? And it's really hard to see where, see this because I'm putting yellow on yellow here. But with these little points, you can make points and they have little numbers next to them. So if you had a bunch of little tiny discrete things on your panel, you could put little dots on them and they would have the number next to them. So you don't have to sit there and keep track of what you've already counted. It's wonderful, right? So these are some of the tools that we use to analyze the panels. So let me pull this up here. Gonna make sure I'm sharing this. All right. So I've created a tutorial for those who are, want to do this kind of work. There's a lovely QR code here in the top corner. Tutorial is also, there's a link. But basically it just kind of walks you through the whole process of using this tool. But what I really, before I get into it, you can do this, that what I just showed you is really good for tunicates, barnacles, a lot of other species. But if you want to look at baby mussels, you want to use AstroTurf panels, because AstroTurf panels have consistency very similar to like the small filamentous algae that baby mussels like to settle on before they metamorphose into their final form. So if you're looking for pesky mussels that settle on things, one thing I recommend is using AstroTurf panels that are about the same size, put in the water, but you can't take photos of these because you can't see the baby mussels from the photos. So what you have to do is you take these back to the lab and then you take a toothbrush and you clean the panel off and then you put it underneath a microscope and count all the mussels. So that's something that I've done in the past, and some months you're counting mussels forever, but you get a pretty good idea of how many mussels show up. So what can be done with these data? This is the fun part, I hope. So there's a couple different tools that you can build, or that one can build, or someone you know can build, that can help you look at your data, right? And there are things called Shiny apps, which are built using our programming language. But basically what they are is a tool where you can upload your own data set. Oh, okay. You can upload a data set and see that data set here, or you can upload an already formed data set, right? And it gives you an opportunity to just kind of play with the data. Now, these don't really have a GIS component to them, right? Shiny apps are good that if you want to share your data and you just want someone to be able to manipulate the data to maybe, you know, look at specific dates they might be interested in, look at specific species, other characteristics of the data, and kind of make some cool graphs to look at. And that might be all you need. But if you really want to use the spatial components, dashboards are the way to go, GIS dashboards. So I'm going to show you some of the ones that I built. These are samples. These are not final products. So here I'm looking at, maybe I just want to focus on barnacles, right? I just only care about panels or places where barnacles are present. So these are my sites. These are the sites that we collected panels for the Salt Kennedy grant. So maybe I want to look at hurricane. It's going to zoom in on the site so I could see where that site is. And I could see that, holy cow, in July, you know, a mean barnacle 7.7 barnacles per panel, right? That doesn't seem like a lot. But if I want to say, look at a couple other sites, then it starts to combine the sites by date. And then we start to see that if we kind of look at all of the sites, July is kind of a hot spot for barnacles, right? There are cases where I could actually play with this a little bit. You could click on specific month and it would zoom in on that month. And so there are different ways you can kind of play around with this tool. Let's see if this one lets me. Are you kidding? Okay, yay. All right. This one is looking at just the percent of biofouling on the panel, right? So what this does is, here's 4 of the sites. If I zoom in on one of these sites, like hurricane, for example, had an average of 99% of the panels were covered. That's that really crazy one I showed you when I did the demo. But if I zoom in on, say, Brewer, right, I could see that, oh, December 1st, 41% average was covered. And again, you kind of play with these so that everything's in the correct order. I put this together just to show you guys some of the stuff you can do. One more I want to show you is, this is with different data. These are data extracted from a white paper. But this particular dashboard, you can choose the date that you want. You can also choose the species you want to look at. All right, so here's barnacles. And if I zoom out, here's the 2 sites that I'm looking at. We can see that of these 2 sites, barnacles were highest in July. But if I wanted to just look at the mussel farm, there were no barnacles present there. So that means all the barnacles were on the oyster farm up here. Let's find one where they're both Batrillus, I think. All right, so we can see the graph updates, right? When I zoom in on the map, that's the nice thing about these dashboards, is that you zoom in on a site you're interested in, all the other stuff that you're looking at, graphs update for that specific location. And that's what makes it really powerful. You're interested in one place, right? You could do all kinds of, you know, neat things with these dashboards. They have lots of different applications. So the vision here is that if you have multiple, you know, growers, for example, sending in their data and having all of these data combined, you could see these overall trends, right? Say over the coast of Maine, or even the entire United States. We were able to get of lots and lots and lots of involvement here. And you can actually see the, you know, overall changes and recruitment for different biofouling species. Okay, this is also useful if you're doing invasive species monitoring, right? If you have a bunch of people who are doing, say, citizen science and looking for a specific invasive species and reporting in, hey, this species was found here, this on this date in this location, you could have a dashboard that updates in real time with the different species that people are seeing, so you can kind of see the spread, right? And be prepared to prevent, hopefully, that invasion elsewhere. So some final remarks for those who are interested in using these kind of dashboards, really, for this specific use, you need lots of good panel photographs, high resolution, the ability to analyze panels for what you want to know. And what do you want to know, right? You need a question. Do you want to look at just hard-shelled biofouling on scallops? Barnacles and mussels are pest species on scallops. Do you want to look at, say, tunicates on mussels? Do you want to look at general biofouling? Do you want to look at just a specific invasive species? So what is your goal? And you also need good data management. You need, a place to organize all the data, whether it's a Google Drive or somebody's got a nice little database, wherever it is you're keeping your data, and sharing it with a central person or group of people who will maintain and update the dashboard or app. So you need some expertise, whether that's something you have in your own organization or something you farm out to other people who can help you with that. So call to action here, because I know there's going to be some folks who are watching this who are growers who might be interested in, if you're interested in having an app to visualize your data or to contribute to a larger community of scientists or growers, I'm your gal. But students who might be interested in a research opportunity to learn how to analyze panels or use GIS tools, I'm also your gal. All right, so there's my contact information there. That's my email, my office number, and my Twitter. So that's my professional Twitter. And so, I'm interested in talking to anybody who, sees a need or sees a use for this kind of tool and wants to learn how to use it or how to contribute to it. And then finally, I want to acknowledge Dana and the rest of the main crew, I call them, but Dr. Damian Brady, his graduate students, and colleagues, and all of the growers who have been a huge part of this, the NOAA Salt and Salt Kennedy grant, and just for this opportunity for me to work with these guys. And of course, I will take any questions. So this is also some wonderful biofouling. This is Damariscata. That's actually right off. This is at the Darling Marine Center. This one's in Rhode Island. And then this is also the Darling Marine Center. So you can kind of see some of the biofouling that occurs naturally. So that's all I have. So I'm happy to take any questions and any. What do you got for me? Anything? We have any questions in the Q&A? Oh. Yes.
So pleased that the GIS would allow you to number the things that you counted, but I was hoping that you would identify a couple and it would count for you. Can it do things like that? Can it help you to analyze your photographs at all? Or is that all still something that the biologist needs to do?
Oh, okay. So that is something a biologist would need to do. So that's just an image analysis program. Now, some of those you can train. I shouldn't say some of those, not that one specifically, I don't think. But what I didn't show you, because I didn't want to run out of time here, you can play with some of the color threshold to just put color where there is stuff on the panel. And this is why you need a panel with like no glare whatsoever. And then you could measure just what, say it's all, I colored it all red. You could look at percent total biofouling, but you would need somebody to be able to identify those species. There's probably a way to train, computers, maybe a little bit, but you really, even there are some species where I'd want to call a taxonomist to be 100% sure, because there are species that look very similar. There's species of mussels that look very similar. And there's a lot of hybridization and overlap. So you definitely want a biologist to identify those. Other questions? We good? Dana, what about you?
I always get something new when I listen to you talk about how these tools are used and what they, how powerful they are. So that was a good review, and then B, there's some new stuff in there. I continue to think that we've got some opportunity there because this is an interest that growers have, and you've done so much work to get all this, the Shiny app and the GIS stuff going, that populating those programs with data, I think, is definitely a possible thing.
Yes.
Yeah, I'm excited by it.
Me too. And they're incredibly fun to design, because it's like you can do so much. You actually can combine all of those dashboards into like one portfolio tool, so you can have them all in one place instead of having to click on a bunch of different links. So there's a lot of flexibility of what we can do with them.
Actually, I do have another question here, if we have time for it.
Yeah, we have time.
All right. So it's basically asking you to speculate, but water temperature is changing, hydrographic cycle is changing. All these things in the climate are hugely dynamic these days. Do you have predictions about, I don't know, winners and losers in the melee of biofoulers out there, or how this community of organisms might be affected?
Well, I tend to agree with those who say that we're looking at a future of jelly, all the gelatinous stuff. I'm just thinking with ocean acidification concerns, maybe not in the near future, but seeing fewer hard-shelled species and more gelatinous species as far as biofoulers go.
Yeah.
I'm also thinking about, species that are kind of expanding northward. A lot of the tunicates that, colonize reefs and things like that, as they are more easily spread. So I'm definitely feeling that the sea squirts are going to win and some of the hard shell stuff might lose. But I mean, again, like you said, it's speculation, but that's kind of where my mind is.
Thank you.
Yep. Thank you. Any other questions? Let me just double check to make sure. Okay. I think that does it for us. And I thank all of you for your attention today at Moss and your attendance. I really appreciate the support, both online and in person. And I hope we can do this again, because, the more we talk about sustainability, I had so many good conversations today with people and students and faculty and people from off campus that I would like to continue those conversations. So thank you again, and awesome. Enjoy your weekend.
Thank you.
Yeah, thank you so much, Dana. We'll be in touch for sure about a lot of this stuff.
Okay.
All right, cool. All right, have a good weekend.
You too. Bye.
Bye.